Okay, I think we're ready to start. Maybe just to go back and re-emphasize sort of the purpose of this meeting and the spirit of this gathering. I think one of from Karsten and from a national science point of view, one of the original goals of this meeting is to try to really identify key conceptual gaps, key technical and technological advances that have to be made to help advance this field. And the other aspect is to explore opportunities for cross sort of collaboration between the funding agencies that are here, represented by Michael J. Fox, NSF, NIH, and CHDI. And I think over the past yesterday and today, we've covered many topics, and I think many people have put their fingers on the right sort of knowledge gaps and barriers to translations. So we have heard about different ideas of what's needed, to move the field forward. So what we'll do here, I would like to start with just a brief overview from each person on the panel, just to say basically, so we have a mixture of companies and foundations. What are you doing? And sort of a quick first impression from the meeting on, for sort of your views on the meeting on these different issues that I just mentioned. So we start with Michelle.
Hi, my name is Michel Steiner. I'm working for a medium-sized biotech company in Basel, Switzerland. We have launched the first product this year. It's an orexin receptor antagonist called daridorexant, indicated for insomnia. And we haven't talked much about symptomatic treatment today. I mean, it's indicated for general insomnia, but I think such symptomatic treatments are very important also for all neurodegenerative disease, and particularly sleep problems and also problems of the sleep-wake cycle are issues that are very relevant for many people suffering from different types of neurodegenerative disease. And I hope there is also funding going into symptomatic treatments and not only into disease modification. And I would wish that also for the agencies to consider. But I guess that's the case. One point I'd like to mention, what I would think is really great in the future, if we think a few years ahead, if we could really identify the different polymorphs and strains of alpha-synuclein, giving an example here, that people carry, that patients carry, and try to match that with the disease progression and the severity of the disease, really to find out first whether alpha-synuclein is really the disease driving factor, which is still sometimes unclear. And second, whether we can stratify these patients and whether we can find different treatments, perhaps for the different polymorphs, if we think of different types of antibodies or small molecules that prevent their fibrillation. So that's the second point. Yeah.
Okay, hi, I'm Ignacio. I work at CHDI. I'm in charge of unintended lowering, and I also fund academic work.
Okay. Kristan Blagoyev, National Science Foundation.
I'm Austin Yang from the NIH. I oversee the etiology and the basic AD biology program. So we support many of the structural and the basic AD research. I have a few slides. So basically, we have a lot of existing program. Actually, that can help everybody. Yeah.
What? Yeah, we have some. I'm Andrew Comer-Cox. I'm from the Michael J. Fox Foundation. I'm focused on alpha-synuclein biology, and yes, we do have money to give out. And I'm also focused on the environmental triggers of Parkinson's disease.
So I'm Peter Lansbury. You've heard what I do, and we can talk about what I did. But I will say, Virginia, speaking of time, the first time I met Virginia, I was like 29 or something. I gave my first talk at Penn. The second slide took a half an hour. And that was you. So don't get on people because of time.
So yeah.
I remember. Like it was yesterday.
Luis Oliveira, Fox Foundation. I'm part of the scientific staff. Funding agency focused on accelerating therapeutic development in Parkinson's disease. We do fund research from, fostering 4 different categories: the define disease, measure, treat, and enable with resources. Yeah, we funded about a little over $100 million last year, spent over, I think, 113 projects. So yeah, we do want to keep the field moving forward.
But Virginia told me last night that our funding comes with too much oversight. We're too bothersome.
Yeah.
DOD and you. Compared to NIH down
Okay.
here. DOD.
We have a different type of mechanism.
So let me, during the, we discussed quite a lot the importance of patient stratification. So I would like to start with Ignacio and Luis, and then Peter to talk about this issue, since all of you sort of have worked on this. So where are we? What are the barriers, and what you would like to see in terms of scientific advances, technology, or things that would enable biomarkers? So maybe tell us in the PD, where are we? Michael J. Fox spending quite a lot of money on this area.
Yeah, this is really an area where the Fox Foundation is really invested. I think it's something that is hampering the development of therapies. Just thinking about alpha-synuclein, that there's companies are targeting synuclein in every way we can possibly imagine, from down-regulating expression to foster protein degradation machinery to targeting aggregates directly, stabilizing them on any way we can think of. There's some company pursuing that therapeutic strategy at this moment. But exactly how we're going to design a clinical trial around that therapeutic approach there's a lot of questions there. And so the foundation, in order to trying to de-risk the field in that regard, we're really investing a lot in biomarkers. I think the 2 main issues are lack of biomarkers of disease progression. That's a big one. And the second one is sort of, with these approaches, how can we find good pharmacodynamic markers in the brain or in the CNS? Mm-hmm. So central markers of target engagement is also a big issue. So we're we're trying in every way possible. One of the assays that is showing some promising is these seeding assays that I talked the other day or yesterday. Yeah. Now the challenge here is, how do we make them more quantitative? And we're supporting a few projects towards that. But I think it's an area where molecular biophysics and biology can integrate, sort of how to solve this issue. So this would be a good area of research for
But my understanding is, even for these amplification assays, they're not good at detecting early Parkinson disease or monitoring disease progression. They're sort of a binary readout.
That's why I was emphasizing the quantitative power. Like, if we can break that barrier, I think at least we can test this hypothesis. Is synuclein aggregation in the CSF a good marker of disease progression? Does synuclein aggregation changes? We know it changes in the brain, so there's a reasonable expectation that we should be able to see it elsewhere. So I think this would be a really breakthrough.
Maybe
in the
to
field.
add to that, there has been a number of studies on applying this assay to skin biopsies and things, and showing some very positive sign also in people with confirmed Parkinson's disease.
That is correct. And obviously the assay works best in CSF at this moment, but obviously CSF has some practical challenges to collect at a very high number in clinical trials. So we're also sort of trying to explore other matrices where this could be easier.
Ignacio, talk about Huntington, and then...
Yeah, not very different from the Michael J. Fox Foundation, only that we're probably 10 years behind them in terms of the number of people and the approach. I think the the patient stratification issue in HD is driven by 2 things. One is there are clear people that progress to the age of motor onset in a way that is not predicted by the CAG and H scores. It's called the CAP score, and that's the GWAS is identified modifiers of progression to the point of being diagnosed with motor symptom. But those modifiers don't entirely explain progression after you become diagnosed, which is the issue that we would like to address at the moment. So a big effort is going to go in identifying people who, on the genetic basis of different rates of progression in different symptoms after diagnosis. That's one. The second one, which is related to what he said in terms of, prodromal stages, and if we're going to plan an early study with a Huntington lowering therapy, which the field is going to go in that direction, you need to be able to enroll people who are going to be at a similar stage in the progression of the disease and identify markers that will change in the course of a phase 3 study, which is 2 or 3 years. Because in Huntington's, if you do a study that is going to be 5 years long, it's going to be hundreds of millions of dollars, and it's not Alzheimer's or Parkinson. So I don't know if people will do a study like that.
Can I just one question here? This stratification that you propose and biomarkers, these appear as an art. I haven't seen a quantification of what actually is needed. When you say you need biomarkers, is basic. What can we measure now? Is the goal of what we need something reachable that they can be defined, or is just going to be an art? We say, oh, maybe we get a biomarker and things may change.
No,
Do we have a real target for that?
No, I think part of the problem historically has been a lot of the biomarker studies are cross-sectional and not longitudinal. And for a slow progressive disease, you need hundreds of individuals being followed year on year in order to define that you have enough of a sample size to do a study. So that's ongoing. I mean, I think it's just delayed.
So can you quantify how many individuals you need, what level of biomarkers you need? Because that's
ongoing.
Is that something that's a possible target, or it's an impossible target?
No, I think it's possible.
NIH has a very large biomarker program.
Right, but what I'm trying to ask you, yes, you have it. But the question is, what do we actually need? Basically, what would you have on your hand now that you say that would work? What level of biomarkers do we need? How many patients do we need? Do we have a quantification of that? What the goal is?
Well, those things are ongoing depending on the modality that you want, whether it's going to be imaging, which is MRI structural is probably the best predictor of progression right now in the prodromal stage, and then CSF, and that's people, neurofilament and other things. But in Huntington's is a bit early. Okay, people have done FDG PET longitudinal after 7 years with 40 subjects. Okay, is that enough in terms of running a phase 3 trial with 500 people? Probably not. So what the field has been slow in doing is longitudinal measurements of enough subjects to understand exactly how to power a study.
So just to add that one more thing, we heard about all the money that everybody has here. So do we have enough money, or how much more money we need in order to achieve that goal? Or that's not a money question; it's a question of people's question.
It's maybe before it's a money question, and I'll let Peter ask. So if somebody was to give you a few millions of dollars to go after a biomarker, or if you have the money to fund, what would you fund to go?
I mean, there's a lot of reasons to have a biomarker. People are talking about progression as if, and it may be true, and as if there's one Huntington's disease. Yeah. And what we want to do is model progression. So, for example, a GBA PD, which is a small slice of PD, the progression is all over the place. And we don't yet know. I showed data from the PPMI studies, which were 550 patients or something like that, that met our criteria. That is where we could draw a line through their progression. We still don't know really exactly who. So I would say in that case, it would be nice to have data on 1,000 patients followed for 3 years with a distribution of genotypes that was. Now, that data is out there, and it's coming, and in PD, it's being privatized. So Neumora is a company that has $500 million in the bank right now and really nothing. But they're really doing deep studies of progression, clinical features. That's the other thing you want to know is, like, which should you be measuring their tremor or should you be measuring their gait? And it's different for different people. Mm-hmm. The other thing that I think there's a danger in that being privatized so 23andMe is doing it. You know, you didn't think your data was being, he didn't think they were doing you a favor by telling you that you're 1/15th Estonian or something. That's not the favor they're doing. They're collecting all that data and they're reselling it, right? And they're now in the drug discovery business. And there's other companies too. So the Decode guys, there's a lot of companies that claim to have 5,000 to 10,000 PD patients followed over time with deep phenotyping, with imaging data, and it's all going to be privatized and it's going to be sold. And so that's not going to be available for everybody. So I think there's a huge danger if that's all sold, right, that we won't have access to it. And
Virginia?
Anyway, I think that, one thing that we have learned a lot is from the Alzheimer Biomarker Initiative. I mean, I would have to say that between Les Shaw and John Trojanowski, I mean, they really kind of push that now. So it's really standard. And also with the Wkshp people even pushing it further. They have a plasma and tau for Alzheimer now. And I think that going down that road and doing plasma and trying to find something for PD, I think that would be helpful. But NIA is doing a lot on that.
Yeah, I'll come back to that, but Brad, can you hear me?
Yeah.
If you can unmute, I wanted to pose a question to you. I mean, we've.
Can you hear me?
Yes. I mean,
Okay, good.
we heard a lot about the use of sort of conformational properties of the aggregates or different strains as a potential way of stratifying or subtyping these diseases. I mean, from the work that you have shown, you've looked at 32 patients, right? And you still see quite a bit of heterogeneity. I mean, and there you looked at sort of typically the fibrils. I would say there is less heterogeneity at the fibril level except for PTM patterns, not structure, compared to oligomers. Now, if you were to expand that study and look at 200 patients, then the likelihood that the number of different strains will increase. Is it really feasible to think of being able just to divide to subtypes simply based on conformations?
I think that one of the kind of frame shifts we have to make as a field is to not think of Parkinson's disease, but maybe Parkinson's diseases.
Yeah.
And not Alzheimer's disease, but Alzheimer's disease is that every single patient I see has their own flavor of things, much like has happened in oncology, right? That there are molecular markers that can, in at least subpopulations, predict rate of progression and also direct therapeutics. And I think that's where we're going to get to 5 years from now in Alzheimer's disease. Part of that question is going to be conformers, I believe, based on our own study, the study from the Colombian kindreds, and then from the study from Cleveland Clinic that just came out in STM, which used different measures of conformers, but in each one showed that the more aggressive clinical disease had a distinguishable set of conformers in tau at least. So I think that's going to be part of the story, but I would be risking ignoring, many of the themes of the past 2 days if I didn't say that people are going to have different inflammatory responses to various measures as well. And the resilience cases really highlight that. Mm-hmm. And so there may well be, a half a dozen different bins that we end up putting people in, that they're going to have proteinopathy conformer, and they're going to have an inflammatory response, and they're going to have some vascular component that may or may not be TTR, but might even be in neovascularization sort of component. And, pick three more that you'd like, some of which will have a genetic underpinning, some of which will have an environmental push, some of which will be just chance. And we need to learn how to measure something that allows us to put people into each of those half dozen or 10 categories, and then ask what the therapeutic that's appropriate for them are. I think Jeff Kelly's experience is so informative. On the one hand, absolutely miraculous that 2/3 of the patients who receive this drug stabilize and improve, it despite even not moving the amyloid out. So really an amazing result. And I think if at the beginning of all those studies 20 years ago, you would have said, Jeff, we got good news and we got bad news. We're going to be able to do, a great thing for 2/3 of the patients, and a third, well, it won't be so great. He would have happily made that deal with the devil. However, it's a cautionary tale, right? What if, what if some of the drugs we're using now really have some beneficial effect? But you saw the spaghetti plots of the, rate of progression. It is absolutely unanticipatable at baseline who's going to go quickly and who's going to go slowly right now. And so I think that unless we find better ways to stratify, we're really, we really are just putting ourselves in a challenging position, much like cancer was in 20 years ago. And so one of the things that I'd recommend in terms of funding would be to look for ways to link neurodegenerative disease research with the kind of thinking and the kind of strategies that have proven so successful in, both inflammatory disorders and also in oncology. And say, how can we learn from our colleagues in adjacent fields and really make those learnings come together and apply them to disorders in which proteinopathies are really, one of the central drivers.
Yeah. So just one, while we're in this issue of conformers, and we always think of these things as a single conformer, but if they were to propagate from one brain region to another, then the aggregation environment changes from one cell type to another. So what if the possibility that within a single brain there may be different conformers or strains, and what are the sort of the implications is for therapies if these are indeed conformationally different, especially for immunotherapy?
We've shown that a panel of antibodies, and we've tested about 20 different tau antibodies now, completely differentially block seeding activity of different individuals, brain material. So, you can extrapolate that to the clinical trial, even if enough antibody got into the brain to be beneficial in, something between 1/3 and 2/3 of the patients. Based upon the assays that we can run in the laboratory, any individual antibody just plain won't work. Okay. And, they're all obviously anti-tau antibodies. So I think your point is really well taken, that we have to do a better job of understanding the conformers and the post-translational modifications that occur in an individual patient before we, maybe take half a step back and see how well what happens in spinal fluid, for example, reflects what's happening in the brain.
Yeah.
Thank you.
So Brad, we actually have done some of those experiments that you just said. So what we did was that we want to ask in Alzheimer patients' brain, are there, in terms of regional variation of potency of the neurofibril rectangle that you get from the same individual? And also, if you now take across, like what you did across individual, and are the potency similar or different? So I have to qualify by saying that the way that we do the experiment is just the way we do it. We extract, the neurofibril rectangle from the brain. And so basically what we found was that from the individual, if you take it from different brain region, there are actually not much difference. Okay, but you have to realize we ground up the brain to biochemical fractionation, just the same amount of total protein and same amount of tau protein. The activity don't vary very much. But from one individual to another, there are variation, and those studies are still ongoing. So I don't have a final answer on how many would do better.
Virginia, that matches exactly with what we've seen as well, and we've only looked at 5 different brain areas. So it's possible that there'd be some surprises if we did a more more extreme microdissections, and similarly, it's a biochemical preparation. So there's a lot of caveats, but that's exactly my impression as well. That Mary's tau is pretty much Mary's tau, and it's different from Sam's tau.
Okay, Peter.
First, I wanted to thank the organizers by saying that I really learned a lot. And I also learned a lot about a sort of different stage of the problem than I was expecting to hear about. And maybe I'll make a remark about that I'd like to hear how the NSF and the other foundations could interact. It seems to me that from the discussion we've had so far, the private foundations are especially interested in the late stages of study of these problems, trying to get a drug and so on. The second you touch a human being, this costs a huge amount of money. That amount of money is more than the NSF will ever get. And so I think that where the NSF can come in, and people like us, is somehow to identify, with your help, those areas of fundamental biology where there's something that's unique in neurodegenerative disease where we can leverage deeper understanding. So for example, aggregation is an example of that. The phase separation problem and things like the postsynaptic density is an example of that. But then there's also all the system biology of the control of the degradation cycles, transport. It seems like there's many, many fundamental areas, but I kind of feel like that's divorced from the considerations that you have here. And I would say maybe take an interesting cautionary tale from Jeff's work. Jeff seems to have worked this out not by starting out trying how do I do clinical trials. He started out by thinking about the problem of how to intercept the process at its earliest stage, which I think is why he's, well, it may be one reason why he's successful. He's also probably successful because he's brilliant and nice and all those other things. But that might be a better lesson than to spend all of your efforts only on the end stages of the problem.
Well, Karsten.
Yeah, so NSF working with NIH very hard. It's very hard because NIH, in my experience, the last 15 years, thinks that this is their land. And last time I worked with Stand Up to Cancer and NCI, we had even a memorandum of understanding, and they pulled out from that collaboration. So I work with the private foundations much better than with NIH, but there is always hope that NIH will. I remember talking to the director of NCI during the Moonshot to Cancer, and I was on the White House working group, and he said, "What do you want? All the money are for NIH." I said, "Why am I wasting time then here?" And it was absolutely true. So it's hard to work with NIH, but maybe with them will be different this time. Maybe NCI is different. So let me just finish. So in terms of the science, so I think that the first thing to identify by listening, so as a complete non-specialist or not knowing anything, my first impression is that we need to find out what causes those diseases. My impression is that there is some uncertainty. Are the plaques, is the tau, and all those things, aggregates. I mean, we might be working on something for another 30 years, and we figure out that this is not causing the disease, or actually the symptoms and so forth. It could be, and I appreciate that idea. I think that we are finding more and more the role of the immune system in all this disease, in cancer, in neurodegenerative, as multiple people said. So it's. The other thing is that it seems to have made progress in cells, but it seems that the problem is like in every other disease that we treat, is the problem, the interaction with our drugs with the human body. And things like delivery and so forth, the fact that some are non-responders, some responders is absolutely the same in cancer. As you know, the immunotherapies for melanoma has about 45% cure, and the rest, 55%, nobody knows why they're not responders. So this kind of stratification is common among complex diseases. And here comes what Hilal was saying, that we need to embrace this as a complex disease, and we need to look at it from a complexity point of view. And in NSF, we have these convergence initiatives that is convergent of multiple sciences, chemistry, physics, mathematics, material science, onto a given problem. And we have done interesting workshops and hopefully funding on cancer, bringing chemistry, material science, physics, engineering as well, computer science. And I think this is the same thing. The way I personally, if you want me, what I would try to do is try to put teams together of different scientists coming and engineers coming from different fields. And with experts in the field of different diseases, and try to have 5 years of these collaborations that are multidisciplinary, convergent onto the problem. Peter and I had a small conversation yesterday that, so I've been trying for 15 years to bring physicists that are not working on biology to think about biology and to develop new tools, to develop new imaging methods. Certainly we need here new imaging methods and new measurement methods to look the progression of the disease. Hilal mentioned that it's different, the process is different from the endpoint. And we need to understand the process. For example, those aggregates, how are they growing? Are they exponentially growing? Are they linearly growing? Are they growing only in the surface? Are they growing in the bulk? How are they? Even this question I think is important. So from my point of view, I will work with the private foundation and with NIH. Try to have some initiative where we can put basic scientists with the...
So if I understand from our discussions a while ago, we are planning this meeting, that there will be mechanisms in terms of building consortia, of bringing people with the right expertise to tackle a specific problem rather than the standard calls, right?
Right. So we have a mechanism at NSF that some of you have participated. It's called Ideas Labs. It came from the UK 15 years ago. It was called Sandpit. And this is where we put 25, 30 people in a room. And like today, but for 5 days. And they form teams. So they form teams and they define problems, and every day they report on those. And people who are the rest of the team, the rest of the room, criticize the team. And then we have also mentors, which are some experts, which are, experts in the field, but more senior. They also advise the teams. And then on Friday, we have final presentations of the 4 or 5 projects. And in the past, we have funded 90% of those projects. And usually these projects of the order of several $1 million per year. And the more money we can collect from, say, NIH, if we work together. I did it, Ideas Lab with Stand Up to Cancer, which seemed to be successful to some extent. But I have done many other ones. And it's a good mechanism because it's not call for proposals where you send, the call is actually for 1 page only. And so anybody can apply. And then there is a committee that selects who to come.
Okay. So this could be sort of, in a sense, this potentially the second phase of this work.
This could
and
be
the
the
idea
phase.
that, we will be reaching out to everybody to try to crystallize a few ideas, and as a second step, there'll be an Ideas Lab meeting.
Right.
To try
And
to
I just emailed today with the people in our international office. So the memorandum of understanding with the Swiss National Science Foundation has been signed. And so now they're working on working plans. So we will have in a few months a document saying that we would like to collaborate. So we could do this at the international level.
Okay. Austin, from your side? I mean, from what you've seen.
I think I want to focus. We're not going to have effect treatment unless we have a better understanding of the disease etiology. So that's basically, Jeff just gave the perfect example. So we need to know better about disease biology. So. But I would like to point out we do have several funding opportunity already exist. So maybe some of you are not aware of what we already have in the pipeline, especially in the area of protein polymorphism. So
and many of these are also open
I will
international?
tell you because so this is already have. And then Brad was on our council member, so he helped us to basically formulate a lot of some of these the concept already. So if I can show you what we have.
If you can share it, go ahead. You're logged in?
Yes.
So the other thing too is that you want to let people know that you have money.
Well, NIH cannot give you money unless you apply. So I encourage.
But there is a lot to be had.
So we do it. So this basically started in 2014 when Brett actually held up the formulas. We really focused to try to capture the complexities, the AD, the basic disease biology. So first year we allocated $20 million. So this project started from 2015, now has been continuing. So essentially, this is basically just to re-emphasize, we really care about the basic AD biology. And then in 2018, we have a program announcement, basically specific target protein polymorphism. So we have more than 10 investigators in this area now. And some of you probably know we already have a grantee meeting. Virginia was there, Jeff was there, Dave Eisenberg, so everybody was there. So basically, we really try to address the issue of the aggregation coming from the structural point of view. And at the end of the meeting, David Eisenberg and Charlie Clay come up with a white paper. So now we're in the process of forming Alzheimer's disease protein polymorphism research network. So last year, we have a tau protein polymorphism network. David's part of it, and we have Jeff Correct and some other tau biologists. Basically, the idea is nobody really know what aggregate oligomer is. Everybody just kind of look at smear on the gel. So ideally, we want to have a better structural definition on what those molecules are. So we're developing the tool for the antibody. We developed a tool on how to make those oligomers. We developed try to standardize all the methodology, how to make all those compounds, because the issue of polymorphism, you can imagine, if you have two labs trying to do the same experiment because of polymorphism, you will get a different results. So unless you have a standard protocol to tell people how you can reliably track these molecules, it's just impossible to address this. So we need to have a standard set of reagents. We need a standard set of a compound, but at the antibody and a chemical probe. So now we're going to form the research network. One is on amyloid, one's on tau, one's on alpha-synuclein. So each center will be supported with a $1 million direct cost. So basically, their job just to characterize the reagent and then distribute those compounds.
Is this US labs only or international? Open to
No, this is open to all the community. Okay. So the lab now has just got funded last year, so they are in the process of ramping up. So David's part of it. So every single compound they use to detect those oligomers will have a cryo-EM, will have a crystal structure. We know exactly where they bind, we know how they bind, right? So this is basically, we're trying to develop a series of standard tools so we can reliably track these type of molecules. Unless we can reliably track these molecules, we'll not be able to understand the disease biology. So this is, we go back to the very basic, the AD, the structural biology. So finally, in addition to our ADR1, I think some of you know that we also support the large, complex, the multi-component project. Virginia and John basically will help them to set up this alpha-synuclein and strain the National Center project, right? So this is things that have been going, and we're very happy that we're able to attack this issue. So not only just making two, but we only want to link this to a clinical component. So you're going to have a large project. There's no budget limit on the U19. You can go up to 5 million and 10 million a year. So finally, this is just one example. We use the multi-omic approach. Now we use the mass spec imaging. So essentially, this is the product initially we just started. So we not only know everybody using the omic approach know what go up or go down during the course of AD, but we know how this thing happen, where do they go up, or where do they go up, because have the spatial information, so also very important too. So that is just a couple examples to what we are doing now, and I think they are relevant to what everybody did.
So Jeff had a comment, I think. No.
promise it'll be really brief. I think Peter Waldness made a really good point that, you know, what I think what we should think about is we need great basic biology understanding if we're going to make innovative drugs, period. And like, let's just take autophagy as an example, right? The genetics are pointing to degradation. It's not only autophagy, but the proteasome. But we don't even understand the basic biology of autophagy. So maybe what we ought to do is decide on a half a dozen or a dozen really basic things that would involve mathematicians, theoreticians, and biophysics, which incidentally isn't really supported very strongly by NIH anymore because you can't get stuff through study sections in biophysics. So I think there's a huge opportunity here for NSF to fill a void. It's not exciting, according to study section.
So, yeah. Peter, I'll come back to you, Judith, because I have a question as well. But I have a question to you first on this topic of oligomers. In 2005, we wrote a review together, if you remember, and the title you had chosen was "Is as good as it gets good enough." Remember? And the editor said we should change it because it will affect funding in the field. So I'm asking you that question again. So reflecting almost 17 years later, would you still use the same title?
I think, yeah, I don't want to comment on. Obviously, this is a great amount of work. It's all in the direction that we could have predicted. I mean, it's just getting more detailed on ideas that we're all convinced are true. So I would like to sort of propose a backwards thing where instead we look at patients, and there are ways to cluster patients. This is what Numora is doing and things like that. They're saying here are a group of patients. Now we take 10,000 Alzheimer's patients, and here are 500 who are similar in the way they progress, the clinical features that progress, maybe in their genetics, and then look at those patients and ask, are some of the, where do you find signatures like this? Now, beyond signatures like this, what I would really like to see from an NSF perspective is more tools for brain imaging, because there's beautiful work in brain sections, beautiful. I mean, this mass spec imaging talk, I just can't get over it. I was texting everybody in my labs saying, you've got to read all this guy's papers. It's beautiful. We talked about this, but it needs, in my opinion, its full power can be unleashed by having a correlate that could be observed in human brains, and then are all those patient subtypes. They're distinct groups. It's not a schmear. It's not like there's a Gaussian distribution of Alzheimer's disease and you're cutting it into slices. There are absolutely distinct things. We've done this with Wkshp, with GBA Parkinson's, and not only is the rate of progression different, but the stages of the disease are distinctly different from Alzheimer's, from typical Parkinson's. And then we go in and ask what transcripts determine that difference. And we found something that was like completely off the wall, totally unanticipated. So I think that there should be somehow to focus on these patient subtypes. Also, they're not doing anything creative with these subtypes. All these companies, they're saying, oh, we're doing deep phenotyping. Well, deep, come on. And they're doing imaging, they're doing standard imaging methods, they're doing standard whole genome, they're doing, it's all standard. What could really turn it into something really useful is if you took those groups and hooked them up to like people who knew how to develop new brain imaging exploratory markers, and then say these two groups that progress very differently are different by this, this marker, and then sort of go backwards from there. Mm-hmm. But you can go also forwards from this.
They're not mutually exclusive.
They're not mutually exclusive at all. But it's like. The problem to me with this whole oligomer thing is at the end of the day, how do you pick the patients who you're going to treat? You're going to have this incredible thing. Oh, this is the antibody I'm going to use for this conformer. I believe. So what are you going to take? So the first 100 people that show up with an ADAS-Cog score of whatever are going to be in your trial. You know, you don't know how to pick the patients. And without that first experiment in humans, it's just going to go to waste, I fear. But if you had an idea, if there were people closing in on both sides, or from the top and the bottom, and now you said, I have an idea that this is an important feature, and this group of patients here, who I can register, and I know how to qualify them, and I know how to find, I know where to find them, maybe even. Those group of patients would be the good people for the first experiment. Then you could do an experiment with 50 people, maybe, and learn something.
Okay, so
very quickly to what
quickly,
Peter was
that's
saying.
fine. And then
Just to say
Judith.
that we at Fox, we do, but like besides the funding aspect of it, we do have, not brains, but we do have from the PPMI study all these biosamples that are accessible upon request to really test these new technologies and to really, dig deep on how to stratify these patients and how to distinguish. Like it's. I mean, yeah, it's only brain. Like we can't take brains from people, at least live people. But we do have all these biosamples, and they're available.
I mean, really, what is the end game of all of this? How do people really imagine that this is all going to turn into something great? I frankly, my imagination ran out in 2005. And, no, seriously, it did, because I thought we're never going to be able to pinpoint that active conformer if we're just studying things in a dish or in a brain sections. It's,
in any case, I think that you need to tell
what
these
is the end
people,
game?
your NSF, the most important thing is that you tell them that we entertain grant submission from you guys, because you're doing basic science.
Yes.
So you fund basic science, you don't fund clinical trials or that stuff, but this is your chance and their chance, so that you can talk to them, right?
Okay. So maybe we'll go to Judith if she's been waiting. Judith, do you want to unmute and speak?
Sure. I actually, by now, I have accumulated too many comments to.
Go ahead.
So first, obviously, the issue, I want to make a comment about the oligomers and the conformers. And sometimes I have the feeling that both with the EM studies of different fibrils and the antibody studies of different conformers, it's a little bit like the guy that lost his keys and looks under the light, because there he can see them, but maybe that's not where he lost them. Because this is based on my experience looking at conformers of Huntington, what you see is highly dynamic, and the oligomers that antibodies detect, or the fibrils that you can capture, are the ones that are a little bit more stable. But that doesn't necessarily mean that those are the ones that are linked to disease. It could be that the cellular environment or the stress conditions of a particular type of neuron favors one over the other, or clears some more rapidly than the other. But it's not necessarily clear to me that targeting one particular type of, I think this is an important question to be investigated, but it seems to me that the discussion kind of like implicitly accepts that some conformers are the pathogenic species, and if we raise antibodies, that will prevent one type of diseases. I also have the same feeling with the following of patients, which obviously this will be very useful, and I would like to see 23andMe making inferences about Parkinson from one DNA sample without having clinical histories akin to the ones that you have in the UK Biobank or something like that. In a way, they don't know the progression of the disease. And obviously, this is also useful because it may help for clinical trials, but there will be no successful clinical trials, in my opinion, if there is no understanding of the basic mechanisms of disease. And I'm not sure that transcriptomics or any omics or any imaging in and of itself will help understand mechanism. It will just identify correlates, but not causes of disease. So I really would like to echo Jeff's comment. We really need basic science of neurodegeneration. We need to use maybe more dynamic, I mean, no, I'm not saying that EM is not amazing, because it is, but we need to use dynamic measures like NMR, maybe non-perturbative measures, to understand what is the dynamic of the oligomers. I think without understanding the problem, you can have all the markers of progression, but there will be no successful clinical trials, because you need to understand the problem before you can solve it, unless you get incredibly lucky.
On the oligomer issue, I would make one statement, though, that we still don't understand what the nature of the native oligomers is. I think Brad's work is beginning to unmask that, and therefore, I think one top priority there is to understand the properties of the native, so that we know that what we can generate in vitro can approximate to what's happening in vivo. And there, I would say that a lot of work, and I showed the data on the screening of the antibodies. So far for synuclein, we have failed to identify a single antibody that is oligomer specific. Yet these antibodies continue to be used, and people continue to make claims about them. So sometimes even whether the oligomers exist and to what extent. So I think we do need to approach that from the basic sort of biophysics, because we can ask questions that you cannot ask with native. But somehow at the same time, we need to put emphasis on understanding sort of what's the nature of that in the brain. I would like to give, I think Herbie had a question or a comment. If not, quick, and then I'll come to you.
So I don't work in this field. Last 10 years I've worked on cancer, and it's been very interesting listening to the 2 days of this compared to what would be the equivalent 2 days if this were a cancer meeting. And I find myself thinking that, with some exceptions, obviously, but just as a generalization, there's 2 points of focus of the committee. There's the molecular focus, molecular biophysics, or whatever you want to call it, the conformers, the misfolding, whatever, which, okay, there's a large group, the people that are working on that, investigating basic questions. Then there's the patient end, looking at real humans and drugs and imaging of humans. And then in a cancer biology meeting, there would be a middle, which would be mouse models, would be cellular studies in detail, not just omics for the sake of omics, but really going in and asking what are the transcription patterns, how is it correlated to cell health, metabolism. So that whole middle ground seems to be mostly suppressed here.
No, I wouldn't say that. I think there is quite a lot. The meeting, we couldn't get everybody, but there is a huge, large number of
but what I see here is trying to go directly from a molecular perspective to drug therapy. Now, that works in some cases. I mean, Jeff proved it works. But it seems like that's not going to work in many other cases. So, for example, no one described to me what's special about different. What? No, I understand that. But okay, so you're telling me the meeting is not. But okay, fine. But then it's very hard for me to understand what are the fundamental issues at the cell biology level, at the model organism level, that really will help the field if this is not.
Maybe you should
okay.
have been invited to this meeting.
Okay, I was one of the organizers. So, okay.
Yeah. I think to be fair, this is,
the brain is more complicated than a tumor cell. Right, and the phenotype of Alzheimer's disease. One of the phenotypes of Alzheimer's disease is the loss of humanity in the patients, and humanity is a complex thing. It's not cell growth.
I just want to give some little bit of hope about antibodies.
Medically, it's similar, but biologically, it's much more complex.
Well, this is enough. You can stay here all night.
That doesn't argue against doing cell biology.
Yeah. But I really just want to make one point, okay? I just want to make one point. There are antibodies that you can make. We have one that actually, if you don't know anything about tauopathies, okay? And this antibody only pick up AD tau, doesn't pick up other tau. So if you're an idiot like me that can't tell the difference between the different tauopathy, and sometimes they're all in the same case, and then you stay with this antibody, you know that it has Alzheimer's pathology. So those type of things is possible.
Yeah. Just on the representation of the different fields, this was a very small meeting that was supposed to bridge people from very different disciplines with a limited number. So to be fair, there are a large number of people who are doing cell biology. It is potentially our fault that we didn't put enough spotlight on that, but we were also limited in time. So I think I'll let Malu speak, and then Sheena, sorry.
Just the point that, the basic biology of normal is not known. And so when you start looking at the aging process and how that contributes, that's not known. So the way that NSF can really contribute is the basic biology of human aging, and then we'll know what normal is. And while there's animal models, we don't know enough about the human immune system to begin with. And we actually don't know enough about the human immune system in the diseases. So we need to bridge that gap, and we need some longitudinal studies.
I'll come back to you. Sheena?
Hey. On Jeff and Judy's comments about basic biology and basic understanding, and I think one of the ways we can link the molecular to the cellular to the model organism to humans is to have much better chemical probes. And I think antibodies are very powerful, but they're very big bullets to go looking for small differences. And maybe the reason you can't find antibodies specific to oligomers is because they may be very similar to the structure of fibrils. Now, we have the structure of fibrils now to atomic resolution. You should be able to design small molecules specific to one over the other, and then build better small molecule probes. And Jeff has used small molecules a lot in the TTR story, didn't talk about. So one of the things I think we really need is to use our biophysics, biology, and we need probes. That's what we've heard about all the last 2 days. What have I got in my cell? What have I got in my brain? What have I got in my test tube? And how do I link it? So I would think that I agree that we should look at the basic biology, and I think we need better probes. And I think actually we're in the right time now to do it. And we don't have to use GFP, and we don't have to use antibodies. We can be much, much cleverer than that. So I think probes, the way to join it all together and understand the biology is through probes. So count me in for probe development. If you can have, if you want Brits. I know, but it's a massive field. Yeah, but it's a massive field. Yeah. But you need probe.
So it brings back things under control. So Karsten. And then Luis, just to respond.
So the chemical probes are fine, but this is like old stuff. I think that what we need is actually responders, and we're developing nano responders from inside cells. These are reporters that are much smaller, they're much smarter, and I think that this is the future. I understand chemical probes are great, but they're not so specific.
I just meant small molecule modulators.
Well, these are not even molecules. These are just nano structures that actually are reported for. They're like small antennas inside cells. This is what we're trying to push at NSF in MPS, and I think this has a big future for.
Luis,
I just wanted to have this small comment, from a funding agency perspective that, I think there's a lot of, I think we did a great job this meeting sort of depicting the complexity of these diseases. I'm not sure if we got to a point where we're starting embracing that complexity. We still see things in isolation. Neuroinflammation has its own, spot, and then aggregation and chaperone, proteostasis machinery. Those are still seem to be operating independently. So I think there's a bridge to cross instead of embracing that complexity. But I want to say something, which is like we're talking here a lot about, we need more basic science or we need more clinical science or what. I don't think we need to choose. Honestly, from my time at Fox, I haven't been in a single Fox internal meeting where we said, we saw this really interesting and transformative project. We don't have the resources to fund it. So I don't think, this never happened and Andrew, I don't think you experienced this as well. So it's really about, trying to get those transformative ideas, because we get a lot of grant applications that are, going again and again over the same problems. there's an opportunity here, and it's not lack of money, I think.
Yeah, and I must say that the Fox Foundation have changed their approach over the years in the sense that they now use the two strategies of the calls plus, receiving input and ideas from people. So
directly, yeah, and we do have consortium, like, for instance, we're working, Hilal is part of that consortium, trying sort of to understand whether the seed amplification assays faithfully replicate, the aggregate structures or not, because if they did, obviously, opens a lot of opportunities for testing ligand bindings and testing the biology of those aggregates. But like just an example, this didn't come from a call. It came from conversations and workshops and meetings like this one. So that's also definitely a possibility.
Can I just say?
Yeah. I'm a theoretical physicist. I don't know everyone we find.
So anyway,
not a lot.
They don't apply.
So let me start a little bit. Christian, let me just make a little bit this point. I see the point here, I think that Christian was trying to make, and I think Jeff made and Peter make, is how do you bring people from a community that's not part of this community into the problem, right? So they're not going to write grants to you, because I don't think if I look on start from my book now, say, do I send a proposal for the Fox Foundation? I think this guy's going to think this is completely makes no sense. This guy doesn't exist. I'm trying to say that's the way people attitude. So how do you bring the people in there? I think that basically the base modeling also involves, I think, today, a lack of theory and modeling. There's data analysis, but there's not real serious theory modeling going. We heard a little bit on phase separation, but there's a need of increase of that into the field. So I think people come from physical sciences. We can be very good at helping people on the service department, do better imaging or do better probes. But I think there's another part that's just taking that attitude. I think Chin is here, and basically 20 years or 30 years ago in protein folding, when there was real connection between the experimental community and the theoretical community, the energy landscape and all these things changed the field of protein folding. So this is something that could happen on coming together. And I think that's something that I think will make a lot of sense for us.
So maybe, if you don't mind, give a mic to Rohit. I put you
No,
on
was that Greg?
I just want to have a follow-up question on this, because you've been sort of one of the people who are at the forefront of trying to bridge biology and computational and sort of basic molecular understanding. So, I mean, you started doing mostly computational, and then you worked, especially now in the phase separation, it's sort of, the two are intermingled. Then I just wanted to get your reflection on sort of how the field have evolved in this direction, how much similar activities are ongoing, and where we can integrate more on the computational side to be able to deal with this complexity. I mean, we've talked about the issue, for example, the topic of post-translational modification, in my opinion, is not something that can be addressed experimentally. Just the combinations, the numbers, this. Yet if I look at computational approaches today, it's hard to find a system where you can predict it. Most protein engineering approaches just ignore it. So, how far have we come, and sort of where do we need to go in this, and what are some of your suggestions? And then I think we'll follow.
So
Similar
I wasn't
question.
sure what spotlight I was being put on here under. I decided to try to be quiet through this because I must admit it got a little chaotic with a lot of what I would think of as, sort of division of labor types of proclamations. And so the approach that I have found to be very productive, at least for my little operation, has been, I think from the theory side, it is really important to understand and speak the language that the experimentalists speak. It is very, very easy to come at, and I mean this with from the bottom of my heart, with all respect to my theory friends and physics friends, whether we like it or not, we come across as being opaque, arrogant, and hard to understand, right? And so that is a divide that I think only we can help overcome by honestly bringing a level of humility to the discourse that we have with our experimental colleagues. So that's philosophy. As far as brass tacks, how do we actually approach something? The central problem of heterogeneity is one that, honestly, the challenge is that experiments have really not been able to address in a way that is clarifying, right? Because most good measurements are exceptional when the systems and the preparations are extremely clean. When you have complicated mixtures, and I'll give you a very specific example, we are fairly confident that a lot of disordered proteins that are also going to be substrates of post-translational modifications have numerous charge states that coexist in solution. Show me one measurement that can actually detect that. So what we've done recently is gone all the way back to Kyle Lindstrom-Lang's approach on potentiometry, but the issue wasn't that you cannot measure. The issue is that we don't know how to interpret something like that. And that, to José's point, required the development of a formal theory that allowed us to do this, right? And so I would say the theme that came out from this meeting is, how do we make measurements in really messy systems that are interpretable? And that is where I think I would fully endorse what José said. The theory comes from statistical mechanics, and that's where we need to think about this more from a theoretical formalism, because the computations aren't really there. Because honestly, a computation will give you one force field, and we'll all basically throw eggs at one another about whose force field is better, when in fact the underlying force field always assumes a fixed charge state, a fixed sequence, very few ions in solution. So these computations are actually super rudimentary still, right? So I think what we need to do is, and this is where I would say the learning methods are really helping build the bridge between experiment and computations. Because yes, we cannot incorporate all the complexity into a simulation, but with the right kind of experiments, with the right kind of sort of detail in the computations, learning can become the bridge, right? And in fact, I should point out something that does get missed a lot. We celebrated AlphaFold in Sheena's talk. Let's make sure that we remember that the genesis of AlphaFold was from the incorporation of direct coupling analysis via something called a Potts model that came from the person sitting to my northwest here, right? I mean, so essentially, the point is that, that was evolutionary information. That was the recognition that there are many, many more sequences than there are folds. So I think this type of information mining and incorporation into building the bridge will really help a lot. And it's doable. It's totally doable. But it requires, I think, a lot more dialogue. It happens right now on the sort of foot soldier to foot soldier level. But I think in a setup like this, very quickly we end up with a balkanization that prohibits the dialogue rather than facilitates it. I don't know how to solve that problem, so I just sort of do it under the radar, but I think it's doable.
Okay. Yeah. I'm a little bit biased in terms of what I'll express my opinion. Last 2 days when I heard all the talks and complexities and the immune systems interacting with amyloid formation and other things, it was clear to me that basically what's going on is that we have systems of thousands or millions of things interacting in very nonlinear ways that it's hard to imagine how a human being can just interrogate one thing in a lab, let's say one protein, one probe, one biomarker, and make any sense of it. I think I sort of see the only way forward is to have system-level modeling. It's not something we can describe with words. It has to be a quantitative model. I think it has to be at the organelle level. It has to be at the cell level. It has to be at the tissue level, maybe multiple immune cells interacting with each other. I think only when those models get developed and then they get validated and become predictive, then we'll have a massive, I think, breakthrough, because then we understand what's going on. Otherwise, it's like super complex, high-dimensional, nonlinear system, and we are just poking it from different sides and seeing different things, getting sort of inconsistent results. And I think in terms of modeling, I've done also lots of modeling like protein folding and then structural modeling. I think there is a large community. We have tools like force fields. That kind of modeling has been going on for 60 years. Not that it's solved, but I think it's more mature, it's more solid. I think at this level of cell scale modeling and tissue scale modeling, it's almost non-existent as a field. And I can't imagine the problems that you are discussing to be solved without that. So I sort of see that as a huge void. and maybe that's one place where there should be some strategic thinking that to bring modeling and experiments to be able to, in addition to molecular modeling, to have also this systems level modeling that could, and maybe a multi-scale modeling that couples both. That's how I see it.
Yeah, so, yeah, go ahead.
So in terms of putting specificity on Garrick's proposal, what I would suggest is that being able to model a process in a cell, right? Jeff brought up autophagy, and I think we know the parts. How do you put a part together that models the output of a system, of a process, right? That's the part where I think the bridge building that Herbie was referring to can actually start to happen, because I think where cancer benefited was there was a very clear outcome. Let's understand what gives rise to metastasis, right, as one example. So in a similar vein, if we say something like, what is it that controls, let's say, synuclein levels inside of one particular type of neuron or tau levels? That moves us in the direction of process control. And that is where I think, not to toot a particular horn, but I think the integration with the emerging theme of phase separation actually becomes very promising.
Yeah. I think the point of this meeting was sort of to highlight those complexity and end with a positive note by showing some of the example of the great science that indeed we're at a point where we can begin to tackle those complexities, right? I think we can tackle them more effectively if we work together and integrate the know-how. You know, when I started working on PTMs, people always told me, which one? You know, if the concept of PTMs on tau, for example, just turns people off because they think I have 45 PTMs to work with, we don't know which one, so just ignore it, right? When in reality, if you look at it at the level of individual molecules, they're probably not a lot, and now we have synthetic methods to generate them and different approaches. So I think in the field, whether it's synthetic chemistry, whether it's system biology, immunology, we're in a great position, and we don't have the luxury because of the lack gap of knowledge to say we should do this and not this. The question is we need everybody to keep doing what they're doing best and move forward, but it's a matter of how we can synergize better and work together better and bring the resources of the different agencies sort of more effectively. You know, if Michael J. Fox doesn't fund physicists, then we need to find this problem where that, you know, sort of where the two can connect. So I just wanted sort of maybe in the closing to ask Austin, sort of Andrew, Luis, and from what you've seen in this meeting and from the portfolio of what you fund and from the discussion, are there specific areas that you can already say we will put emphasis in, or we you know, you already recognize that you want to since you're you funded this meeting, you get to start.
I definitely am looking forward to putting theoretical physicists to encourage them to work actually with teams in experimental.
Okay. Austin, I mean, you talked about I'll come back to Jeff, but you talked about sort of, you provided an overview of what you fund. But
well,
from what
this
you,
is just a very small portion of what we are paying now. I think the Eliezer give a very broad overview on what we are supporting, right? All the way from the basic, the AD biology, structural biology, all the way to the clinical trial. So we support a very wide range of basic, translational, and a clinical study. And
okay. So you're saying that you already have the mechanisms to cover all these different domains.
We have a lot of mechanisms to cover pretty much everybody talk about here. And it just, and NIH really is our primary fund. It's to support the investigator initiator the award, right? So basically we believe
yeah. So Andrew?
They need the multidisciplinary groups though, but so they have the funding, but we need the multidisciplinary groups.
We have that. We have the, not just the R1, we have the U19 with PPG with some more
It's okay, Andrew.
projects.
Okay, Andrew?
Yeah, I think just from my perspective, in terms of bringing together the theoretical physicists and the experimentalists, yeah, I guess to back up a second, I mean, us at the Fox Foundation, we're the largest private funder of Parkinson's disease research, but our funding levels still pale in comparison to the NIH. So we try and we look at what they're doing, we look at what other groups are doing, and then we try to strategically fill in the gaps there. So we don't want to kind of be duplicative in our funding, we want to be complementary. But yeah, in terms of the theoretical combining with the experimental, I think the one thing we've seen is that, I forget who was talking about it, but the experiments and the modeling is only good as the data that's been generated. And I mean, we're still talking, there's still some very basic things about the diseases that we don't understand, that we don't have the data to model accurately. So then we're not sure if we're building these models, investing in models based on incomplete data, then we're not really clear on how good these models will be. So yeah, I think now...
So let me put you on the spot. How much, what percent? Yeah.
The question is on several areas that we worked. When you say data and the experiments, is when the experimentalist and theoretician work together. Sometimes the data you want is not the data that people are being generated. So basically, the protein folding again changed a lot by people coming together. Which kind of data you wanted changed from what the data was. So it's really a two-way road. It's not a way that modeling can only happen depending on how good is the data. It's really a two-way road that goes on this way that needs to be worked.
Yeah, and I think we are enthusiastic about bringing these groups together to get these kind of projects going. And yeah, I mean, it's not a straight line. It's a constant feedback loop in terms of generating the data. Yeah. I mean, you'd have to talk to the experimentalists on that one.
No, but that's why the combination, this evolution changes from both sides what the data you want.
Yeah, I think for us it's still a challenge also in getting the groups in the room together and to speak maybe the same language. And we found that forcing people together into a room rarely works. These partnerships have to happen organically, but it's difficult for these partnerships to happen organically.
So maybe
in defense of the Michael J. Fox Foundation, let me just say, because I've been working with them for 20 years. They gave us our first grant in 2003 on the role of inflammation in Parkinson's. So they've always been ahead of their time. They fund the innovative stuff, and you have a forum for the multi-sector, multidisciplinary collaborations that it takes that they just, they could partner with us, and we know about the PD and the animals, and they know about the theoretical physics. Happy to do that. I think that's the forum. Yeah. And one last thing, because I need to go. I invite everybody to go to the Gordon Parkinson's Conference in Switzerland, Mother's Day weekend, 2023, so we can have more of these discussions, because this was really fantastic.
So before we close, I want to really thank Brad, Judith, and Sarah, and people who joined us, despite, the challenges, and also give them the chance to have closing comments. So I would start with Judith, please, and then Sarah, and then Brad. So Judith.
Before we continue, no. I won't give Teresa a chance to speak. She's trying to speak for a long time.
I didn't see her hand, sorry.
So Teresa, just have the word, because basically she has been working with Creston a lot into the NSF, put these things together, so just give her a chance.
I think that since we are the funders of molecular biophysics, I think there's a lot of things, and having worked in the field before I went to NSF, there's a lot of things that have happened over the last 2 days that are near and dear to my heart. I really agree that a systems approach, whether, designing, or we need to enable the development of new tools to do the, to follow in time courses all of the imaging data, to do it in vivo as opposed to just in the culture dish. But we need to be able to help develop those tools. I think that those modeling approaches that combine the new experimental data that you should be able to collect, if we enable the development of those tools, along with, that you'll get a systems approach of what's happening in healthy aging brains, so that you can also then think about what's happening in diseased aging brains and what happens when we get this loss of homeostasis, if there a tipping point that leads to this cascade of diseases. I think that all of that is space that, NSF could play in, that is at the edge of where we interact with NIH and how we cross into the disease space. I leave that to Creston to negotiate, because but I think that there is a place where we can support a lot of the basic research that is really going to be foundational to enable you to make those advances that will lead to the clinical trials that lead to the new discoveries of cures for, hopefully, or things that mitigate the progression of the Alzheimer's and other diseases of aggregation. So with that, I'm going to stop now. But Ben, thank you. It was a great workshop. Glad that you all were able to participate.
Thank you. Judith?
Well, first I want to say I'm really sorry I wasn't there and didn't get to talk individually with you, but nature intervened. I actually think that, to me, this was really very useful because it led to many discussions of topics that normally we don't think about. I think the problem of neurodegenerative diseases in general has been that we have been very constrained by our models and hypotheses, and it is taking such a very long time to be convinced that they are just not correct. And I think it's taken very expensive failures to bring us to the drawing board to re-examine maybe our original hypothesis of what's toxic or what the disease is like is not correct. And maybe it would be important as a cautionary tale to not fall into other dogmas or hypotheses that will take many decades to figure out are not correct. I think this symposium was very good because there were so many new ideas, and I think it would be good to let the ideas and the data guide the hypotheses rather than try to find a cure pronto before we understand the problem. Anyway, I'm not sure if that makes sense.
Sarah?
Yes, I also wanted to say thank you. This was really fantastic. I think the topic, the theme, really trying to embrace complexity, it was really great to be able to hear that from different people, different angles, different tools, different technologies. I think this was a really good attempt to try not to, I remember we were discussing not having like the full spectrum from brains, people looking at brain pathology, to the cellular model, to looking at the actual molecules. But I think this was a good attempt to have a small enough group for like intimate discussion and to identify areas that we really would need to work more on, and how we can accomplish that, and echoing some of the sentiments that were expressed about having really bringing people together from diverse fields and diverse expertises from, really things not just from biologists, but from, like engineering, from these other aspects. And then in addition, I think also echoing what I believe Brad was mentioning also, is learning from colleagues from adjacent fields. I thought that was really great that he mentioned that, because that's exactly what we're doing with cryo-electron tomography. There's a lot of advancements in pushing resolution in when people are looking at yeast or completely different systems that we can really benefit from applying to neuronal systems and neuronal cellular systems. So I think I really took away a lot from this, and I think this was really a really fantastic workshop. And I hope we can have a longer.
Thank you. Brad?
Yeah, thank you so much.
Brad?
Yeah, I just want to again thank you and join my colleagues in apologizing for not being there personally. If nothing else, it sounds like dinner was a blast, and the workshop as a whole was just fascinating. So this sort of collision of different fields and of different perspectives, I think is really the fertile ground for creative creatively going forward. And so I really appreciate having had the opportunity to participate in this one. And I'll close with a vote that this isn't the last one of these.
Yes. Thank you very much.
Thank
So
you,
I think
everybody.
just to in terms of wrap up and follow up, I think there are two practical steps. One is we need to plan the next step, which will be this ideas lab.
Well, this is up to me.
Yeah, but the idea is in order to sort of potentially make that possible, we will need input from the participants. So we will be reaching out to you by email for some very brief input in terms of trying to crystallize specific fields, topics, and ideas and questions that we hopefully can narrow down into the most important things, or also share with the different colleagues from the different foundations and see how we can move forward. I thank you all for participating, but before we leave, I have to thank Sarah for without her, none of this would have been possible. She's done an amazing job at every step along the way. Thank you.
From Kelly from the
And have a great day. Thank you very much.